The next few slides are slides that help you organize your notes a little bit. They ask you what to think about when you learn a new cell or things along those lines. So I'm going to skip over those in the video. Feel free to look at them. And if you don't have the slides in front of you, you can pause as I go through them to take a look. But what this video mostly will be about is parts of the innate immune system and how they work. So you can pause here if you want. You can pause here if you want. And then if you want, you can pause here with humoral versus cell-mediated immunity. We might come back to this later in the term, or we might not. But the main topic of this video is going to be parts of the innate immune system. And so, okay, so the innate immune system has three broad parts, we could see it as that. Physical barriers that you could pretty quickly name. Chemical barriers, that's molecules we release that protect us in various ways, and then cells that do a lot of different work for us. So this video, we're going to look at the physical parts and the chemical and, humoral parts. Humoral refers to molecules that are dissolved in blood and lymph. So the major physical parts of the innate immune system are going to be skin, which just by itself is a barrier. But in addition, it does something for us. It pushes cells from the stem cell layer here in the epidermis superficially towards the skin surface. And what that means is as they go and they mature and then they become keratinized and eventually they fall off and they take with them anything that has landed on our skin. So if we touch something dirty with lots of scary bacteria on it, eventually that skin will just fall off and those scary bacteria will fall off with it. So that's something impressive. And then our mucous membranes are impressive. They release a lot of molecules that protect us, but just by secreting mucus, they trap potential pathogens, especially in our respiratory system. They trap pathogens, and then the respiratory epithelial cells, the ciliated epithelia, push that mucus up towards the pharynx where we swallow it, and it goes into our stomach, and the stomach acid kills whatever's in it. That happens all the time, whether we think about it or not. And so that's a really powerful physical protection we get. There are others you could name, but these are the ones that I want you to specifically picture. Chemicals typically fall into antibacterial, antiviral, maybe antifungal. And I want to go through the examples of these because they connect to other things we've learned in the class. So for example, lysozyme destroys peptidoglycan. Well, pause the video and remind yourself, where is peptidoglycan? Another thing you might ask yourself is, would this work against a gram-negative bacterium? Answer, no, because it can't get to the peptidoglycan layer. So if that doesn't make sense, pause the video and go look at the structure of a gram-negative bacterium. Defensins are a large group of antibacterial molecules that work in different ways, and they're proteins or peptides. Peptide is another word for a protein, generally a short chain of amino acids. This one is amazing. Dried sweat. What it does is lower the water activity of skin. It turns skin into a desert. So when sweat evaporates, it leaves behind salt. Most bacteria can't survive there. And that really does limit what can be on our skin. And then one that I brought up when we looked at microbial lifestyles was lactoferrin. And all it does is bind iron. It scavenges iron to limit iron available to potential pathogens. So at our mucous membranes, we're releasing lactoferrin to make sure that if there's any free iron, it's caught in this protein. And most bacteria can't get it. And if they need iron, they can't get it. Real pathogens have to be able to steal the iron back from lactoferrin, and they are capable of doing that. Antiviral chemicals make more sense when you have looked a little bit at how viruses work, but take my word for it, some viruses have DNA genomes, some have RNA genomes, and so we have enzymes that destroy RNA and DNA. Your skin, for example, has RNA degrading enzymes that make it very difficult to work with RNA in a research laboratory. It's very difficult to extract and study RNA without accidentally destroying it, because the enzymes on our skin are very good at destroying RNA, and RNA is also unstable. And then the last antiviral chemical I want to think about is not something that directly attacks viruses, but something that changes our cells' behavior and prepares us for viruses. So interferons are signaling molecules, and when other cells detect them, they turn on antiviral responses. And that is very much part of our innate immune system. When we are infected with a real virus like influenza, or if we have a significant infection with like a coronavirus, so generally something beyond a common cold, something like influenza is the best example, that makes us feel crappy all over or gives us pain in our joints. Some of that comes from interferon. The interferons tell ourselves, protect yourself from viruses. Stop doing your job. Instead, protect yourself from viruses. And we feel that. There are other reasons you feel crappy when you have a viral infection. Plenty of reasons, which we can talk about later. So water activity, again, skin, water activity. Interferons, this goes through exactly what they do. When cells detect interferons, they stop translating their RNA. So they stop making proteins. So how does a cell do its job when it's not making proteins? Spoiler, it can't. Instead, it just protects itself from viruses. And they are able to degrade viral RNA. They can usually identify it and destroy it. That was the physical and chemical barriers. In the next video, we will look at the process of phagocytosis.